Well, welcome to all of those who are joining us again for our AMS monthly seminar meeting. I am Jonathan Plett. I'm the president of the society, and it is my pleasure today to welcome Hagai to give our seminar today. Now, Hagai is a plant ecologist from Israel. He is currently doing his sabbatical over here. Originally, he did his PhD at Ben-Gurion University, studying plants' ability to predict future conditions, very smart plants. And he later continued to do his postdoc in the Technikon, studying pollination and genetic limitation as part of the effort to rehabilitate populations of endangered Iris lorettii, which is more commonly known as the Sumerian iris, which I found interesting, was noted in Science in the early 1900s as the most beautiful iris in the world. So since 2011, Hagai has had a position at the Tel-Hai College where he continues to study plant ecology with an emphasis on ectomycorrhizal ecology. Now, Hagai has published some really nice work in the recent years in this area, not only looking at how ectomycorrhizal fungi can promote host growth, has been reported before, but now he's looking into, how competition and abiotic stressors can heavily influence this benefit, which I think is really critical knowledge as we are trying to see in forestry around the globe how to leverage these beneficial fungi. He also published this year in New Phytologist, a 99-year project looking at ectomycorrhizal spore viability and longevity. And I was just super impressed. That takes some immense planning and is generating some really critical knowledge as to how long these spores can hang around in the soil column without a host. But without further ado, I'll pass things over to Hagai to give us unique insights into ectomycorrhizal fungal ecology.
Okay, thank you very much for that very flattering introduction. I hope I can live up to the expectations. I wanted to start with this image. I know that science shouldn't be political, but Israel democracy is at stake, and I feel like a deserter for leaving, and all my friends are in the streets marching for democracy. So I just wanted to start with this saying that you should never take democracy for granted. And that's it. And I want to start my talk with a disclaimer and just to make it clear that I am not a mycologist. And I'm a plant ecologist, and I'm really interested in how EMF affects ecological processes. But even though I'm not a mycologist, I made some very good mycologist friends, which are my collaborators, and everything that you're going to see me present today was done with them. One of them is Tom Bruins, which you might know. He studies EMF ecology for many years. And the second one is Stav Livne, and right here in the middle. And she is my academic wife, and we do all of our research together. And if you ever see a presentation by her and you see some slides that look similar, so I stole from her and not vice versa. So when I was thinking about how to structure my talk, I decided I would structure it around scale, and both time scales and spatial scales. So I'm going to talk about 4 different combinations today of short and long time scales and small and large spatial scales. And I have like one slide explaining what ectomycorrhizal fungi are. Since this is the Australian Mycological Society, I assume that most of you know what they are better than I do. So these are fungi that live on roots, and they have a symbiotic interaction with the plants. In general, the plant gives them carbon, and they give back nutrients, and maybe also water, and maybe also other stuff. But what's important here is that this is an obligatory interaction for many trees. It's also obligatory for the fungi, meaning that they need each other in order to grow and develop. So I'm going to start with short time scales and large spatial scales. And I want to start by talking about this movie, Castaway. I don't know if you saw this movie. It's really good. You should check it out with Tom. Everything with Tom Hanks is very good. But in this movie, Tom Hanks gets stuck on an island with this volleyball named Wilson, and he has a hard time surviving at first. This is what the island looks like, and there's a lot of coconut trees on the island, and Tom eats the coconuts and he uses them to store water. So I think that we can assume that if another coconut was to disperse from a distant island and arrive on Tom's island, that will not have a big change on Tom's lifestyle on the island. But let's say that a porter potty was to disperse somehow and arrive on the island, that's probably going to have a very big change on his lifestyle, and not for the immediate reason you would think of, because in the movie, he takes this porta potty, makes it into a sail, and then he sails this raft back to civilization. So the point I'm trying to make is that not all dispersal events are the same. Some dispersal events are more influential than others. And what makes a dispersal event influential are a few things. One, that the arriving species is new. When you get more of the same, it could be important, but it's usually less important. And if you want a dispersal event to be especially influential, it should provide functional novelty. And a good example for functional novelty is the introduction of predators to islands. Okay, so this is Australia, so you guys have a lot of experience with influential dispersal events. Okay, so when you bring a predator to a habitat where there were no predators before, that's a very influential dispersal event, and it can have long-term ecological and evolutionary significance. So dispersal could have a lot of influence on the ecological community, but when you are an obligatory mutualist, dispersing is hard. Well, it's harder, because it's not only you that needs to disperse, you also need your partners to disperse as well. So both symbionts need the other symbiont to be there when they arrive, and this poses a chicken-egg paradox. And usually this paradox could be solved in a few different ways. One is that both mutualists travel together. That in the case of ectomycorrhizal, that's usually not the case, because usually plant seeds don't travel with the EMF spores, ectomycorrhizal spores. So the other solution to this paradox is that one of the partners is able to be dormant and just wait for the other partner to arrive. When we're talking about tree seedlings and ectomycorrhizal fungi, tree seedlings are kind of spoiled. They don't like waiting for someone else. And tree seeds are usually don't have a lot of longevity. For example, oak acorns die out within a few weeks. They cannot wait very long, and pines are kind of the same. So it's the fungi that have to wait, and usually this happens in the spore stage. So ectomycorrhizal fungal spores are able to be dormant in the soil for long periods of time, and there you're waiting for their mutualists to arrive. And the way that these spores are dispersed is either by wind or animals. And when we're talking about subterranean fungi that have subterranean mushrooms, so wind is not a big factor and it has to be animals. And we wanted to look at this within our system. So like I said, I live and study in Israel, and a very big and influential mushroom and fungal disperser is the wild boar, which is native to Israel. And they're influential because they eat a lot of mushrooms, both below and above ground. They're large mammals, meaning that they have large home ranges, they walk long distance, and they have a long retention time. Retention time is the time between you eat and between you defecate feces. And that's usually correlated with body size. And they also use diverse habitats. So they use ectomycorrhizal habitats. When I say ectomycorrhizal habitats, I mean habitats that are dominated by ectomycorrhizal host trees. And they all, like mixed pine oak forests. And they also use open grasslands. So they move around, and therefore they have a good potential to transfer spores between habitats. Sure. So our research question was, how do spores that are present in the wild boar feces influence the EMF community at different habitats? And what we were asking is, are these influential dispersal events? And we hypothesized that in general, wild boar feces should increase the fungal diversity on pine seedling roots, and that this addition should be more influential in habitats that lack EMF hosts, for example, grasslands. And we further hypothesized that the feces will mostly add species that have below ground fruiting bodies. So in order to test this, what we did is we did a bioassay. We collected soils from the two different habitats, and we collected wild boar feces in the wild, and we autoclaved half of those wild boar feces, and then we grew pine seedlings on the different soils with the different feces. So the autoclaved feces were just a control in order to control for the nutritional effect of the feces. So just really quick what we did, we collected soil and feces, we planted the pines, and then we let them grow, and we harvested the roots, we amplified DNA, we did next generation sequencing with Illumina, and then we did some bioinformatics and analyzed the data. So I'm going to show the data, and what we can see here on the x-axis are the 4 different treatments. So it's the 4 different combinations. So the control is actually these 2 groups, because this is like what the forest community looks like without the addition of the live inoculum, and this is what the old field looks like without the addition of the inoculum. And what we can on the y-axis, we can see the ectomycorrhizal species richness, and we can see that the forest community is richer than the old field community, and this is not very surprising because there are EMF hosts in the forest and they're not present in the old field. And we can see that when we add the live feces, there aren't very big changes to the EMF community in the sense of species richness. So the changes are not that big. And so what we can take from here is that the forest is richer than the old field. But when we look at the community structure, we can see that these 2 communities are very different. So once again, over here, we can see the control. This is like the natural community without the feces. And we can see that both communities are very highly dominated. One of them is dominated by Geoffra, and the other is dominated by Suillus. And I'm going to go into their ecology in a little bit more depth in just a few slides. And when we add the live feces, we can see that this dominance is reduced, but is still maintained to a certain extent. And we can see that the main thing that is added to the community when we add the feces is the tuber. And we can see that the shift in the structure of the community is much more dramatic in the old field community than it is in the natural forest. Okay, we can see here that this is the tuber addition, and it's a significant addition, but over here in the old field, the change is much more dramatic. And when we did an ordination of these 4 different combinations, we got a very nice separation of the 4 combination. And what we can see is that the old field is characterized by Suillus, the forest is characterized by Geoffra, and the live feces are characterized by tuber. So when we look at those different species, they have pretty different ecology. Suillus dominates the old field community. It's wind and wild boar dispersed, but it fruits above ground. It's part of the suilloid group, which is known to have very resistant spores that can last in the soil for a long time. I'll talk about that later on in my talk. And it's a pine specialist. Okay, so it doesn't interact with species outside of the pine genus, with very small exceptions. Geopera, which dominated the forest community, when you look at the soil, when we analyze the soil, Geopera makes up a very small fraction of the soil EMF community, but once the seedlings start to grow, it dominates the seedling. And very little is known on its ecology. It's not a very known species. It shows up in the dry habitats around the world, which are Israel. It shows up in Arizona, in Eastern California, and in Greenland. And the last species, tuber, seems to be the main species that is moved around by the wild boars. It has underground fruiting bodies, and wild boars are known to be very good at locating those fruiting bodies. And it's also known to colonize oaks. I mean, it's famously known to colonize oaks. So it seems to us, at least, like our interpretation of the results are that in different habitats, different ecological processes determine the community structure, the EMF community structure. In the forest, the fungi are able to grow. Unlike in the old field, where it's just a dormant spore bank, in the forest, the fungi are actually actively growing, and there is mycelium, and therefore they're also able to compete. The fungi compete between each other, and it seems that competition could be an important driving factor in the determination of the community. And it's possible that Geopera is just a very good competitor, and that's why it dominates the forest. And by the way, when you collect soil samples in any pine forest in Israel, and we have pine forests from very diverse, under their very diverse ecological conditions, Geopera is always going to show up on the seedlings, and it's almost always a very strong competitor. In the old field, there are no hosts, and therefore there is no possibility for competition because spores don't compete. Spores are dormant. And it seems that the processes and the characteristics that are important for determining the old field community are spore longevity, how long they can survive in the soil without interacting with the host, and spore dispersal. So different processes in different habitats. And maybe Suillus is good disperser or survivor. This is not a very big maybe, because many studies from different places in the world have shown that suilloids are good survivors, and studies from South America where Suillus is invasive have shown that it's a very good disperser. So what do we speculate about the results that we got? It seems that boar feces can effectively disperse EMF spores. And whether these are influential events, it seems like this depends on the habitat. In our study, in both habitats, the wild boars brought in new species, which should be accredited as an influential dispersal event. But it seems like they're more influential in the old field because the changes to the community there are greater. About the functional novelty, so that takes even more speculations, but grasslands seem to be dominated by a pine specialist, which is Suillus. And in Israel, a lot of our pine forests are mixed pine oak forests, but the pines are much better at establishing outside of the forest, maybe because it's full of Suillus, and the oaks are not so good at establishing outside of the forest. And maybe when wild boars disperse tuber outside of the forest that could assist the establishment of broadleaf trees outside of the forest, and that would be a functional novelty because that could have dramatic changes to the environment. So my take-home message from this part of my talk is that next time you see a pile of poop in the field, you should ask yourself, is this another coconut or is this an influential dispersal event? And the second part of my talk is kind of originated from the poop experiment. And when we were collecting the wild boar feces, we were contemplating on how we should apply it to the pots. So one possibility was to take the feces intact and just bury them in the pot. And another possibility was to grind them and then mix them into the soil. We decided to grind them and mix them into the soil. But as we were talking about this, we started thinking about the importance of the spatial structure of the EMF community, spore community in the soil. And then we said, okay, let's do another experiment. So now we're talking about very small spatial scale and also very short time scales. So the question that we wanted to know is how does the spatial distribution of the spores in the soil affect the structure of the EMF community, and how does that affect the performance of pine seedlings? So what we hypothesize is that heterogeneous distribution, which is what we see here, it offers spatial refuge from competition. What does that mean? It means that the weak fungi are not outcompeted because they exist in a place where there is no competition. So they are able to interact with the plant. And we thought that when this happens, we'll have higher EMF diversity, and then we'll have complementarity, which means that different fungi could give different resources to the plant. And then we can have improved seeding performance. In general, in ecology, it's a pretty, well, I wouldn't say established, but pretty accepted dogma that increased diversity should increase performance. So we want to look at that. So we collected soil inoculum, which is just forest soil from 4 different forests, a pine forest, 2 different oak woodlands, and a eucalyptus grove. The eucalyptus are obviously not native in Israel, but we wanted something very different. So that's what we did. And then we had 2 treatments. It was either homogeneous or spatially structured. Okay, so we added the inoculum and mixed it into the soil, or we added the inoculum in 4 different patches within the pot. So what we found was that when we are looking at the community structure of the fungi on the roots of seedlings, when we mix the spore bank, okay, these are 4 different forest soils mixed together, we got very strong dominance of Geopera, once again. But when the soil bank was patchy, we got a more diverse community or a more even community. And notice that these guys are Australian. These are not native. These are native to Australia and not to Israel. And these species, which dominate the pines under spatial structure, they probably arrived with the eucalyptus saplings that were brought probably around 100 years ago to Israel. So when we look at seedling performance, this was like a more surprising result. We see that the homogeneous treatment, the seedlings were more than twice bigger. And I remind you that our hypothesis was the other way around. We thought that heterogeneity would increase diversity, and diversity will facilitate complementarity, which would make the plants bigger. So it was a nice theory, but it wasn't true. But so higher EMF diversity did not result in improved performance. And at first look, it looks like the diversity productivity dogma is at risk. But when you look a bit further into the results, you see that it's not really, that it's a little bit more complicated. And it's a question of how you define diversity, at the plant level or at the treatment level. So we can have a seedling that has 3 different species of fungi growing on its roots, and we can see them right here. And if all the plants in the treatment have those 3 different species, so they all have the same diversity, which is called alpha 1. Alpha 1 is the diversity at the seedling level. And also at the treatment level, they will have the same diversity, alpha 2. Okay, so every seedling has high species diversity, and diversity will be high both at the seedling level and at the treatment level. Another possibility is that every seedling is very highly dominated by one species, but different seedlings in the same treatment are dominated by different species. So this is what we have here. So alpha 1 in this case is low, okay, because the species are highly dominated by one species, but alpha 2 will once again be high because when you average it on this whole treatment, you get high diversity. So here each seedling is dominated by one species, but diversity is high only at the treatment level. And what you see here on the screen, this is more representative of what we got. In our case, alpha 2 is greater than alpha 1. And what it means is that even though it looks like the patchy distribution of spores resulted in higher diversity, that was diversity between seedlings within the same treatment, not within a single seedling. Okay, so patchy spore banks resulted in differences between seedlings. And then we can ask ourselves, what is the mechanism? And it looks like in the mixed treatment, when we were mixing the soil, Geopera was everywhere, and Geopera was always winning and dominating the plant. But in the patchy treatment, where there was some level of randomness to the direction that the roots grew, we had diversity between seedlings. Okay, as the roots were exploring the soil, it looks as if the first fungi they met grew and dominated the root system. So it looks like we have a priority effect, and priority effects have been shown to occur in the roots of pines being established by EMF. So a priority effect is a case where the structure of the EMF, in this case, the structure of the EMF community is determined by the first fungi to colonize the roots. And there are different mechanisms that could create priority effects. It could be the time to germination, that whoever germinates fastest is the one to dominate. Could be mycelium growth rate, whoever has the fastest growing mycelium, or the exploration strategy, the fungi that has the best exploration strategy dominates the root system. So it seems that the spatial structure of the spore bank can interact with these different mechanisms and have an effect on the community structure of the EMF on the roots of pine seedlings. And this seems to be important for seedling growth because, like I told you, we had like more than a 2-fold increase in growth. So we had short and small and short and large. And now I want to move on and talk about long time scales and relatively small spatial scales. And long time scales in general are always neglected in ecology. And the reason is that humans only live 100 years if they're lucky. And long-lived organisms pose a lot of challenges to study, and grants usually don't last for 100 years. But long-lived organisms also spur opportunities because you can discover things that you wouldn't see on a short time scale. And when we discuss organism longevity, so longevity has always, fascinated people, and the organisms that can live for very long are very interesting. And over here we can see all kinds of different animals. This is the longest living animal. Okay, and it's this very small shell, but they live the longest. But so these are all animals. But when you go to plants, it jumps up in an order of magnitude. And I was looking today online for the longest living plant in Australia, and I think it's this. I didn't do an in-depth research, but the Huon pine, which is the common name, is thought to be able to get to around 3,000 years old. And until about 50 years, the world record, a bit more than 50 years, the world record was 3,200 years. And this is a giant sequoia. And these are the biggest trees in the world. They grow in California. And they were also thought to be the oldest trees in the world. But they're not. And I want to tell you a story about the oldest trees in the world and how they interact with fungi and how that could have an impact on their ability to cope with environmental changes and global warming. So I will give a really short overview of how they understood, how it came to be discovered that these are the oldest trees in the world. And then I'll tell you about our research. So it happened by chance. And this guy, Edmund Schulman, he was interested in ancient climates, and he was looking at dendrochronology, where you drill through the tree and you count the rings. And he was looking for old trees. And at the time, everybody thought that the sequoias are the oldest trees, and sequoias grow in California, right here in the Sierra Nevada, which is the big and famous range of California. And at some point, Schulman met up with a forest ranger that was working in the White Mountains. These are the White Mountains, this little range here. It's not that little, but it looks little compared to the Sierras. And it goes up to 4,000 meters in elevation. And it's a desert, but it's very cold desert. And most of the precipitation comes down as snow. And he told him, listen, this was in the 50s. He told them, I've been up there and there are some really big trees and weird looking trees. They look pretty old to me. And he was talking about this tree. It's called the Patriarch. Okay, this is a Bristol cone pine. And these trees dominate the tree line of this ridge. And this specific tree was very large, and he told him that he should go and check it out. This is me and my son checking out the exact same tree. But so Schulman at the time, there was no roads going up there. He had to find some students and hike up the mountain. And then they established camp, and they found this big tree and they sampled it, and they discovered that it was only 1,500 years old. And they were very disappointed. But they walked all the way there and set up the camp. So they said, okay, let's stay around and look at some more trees. And they looked at other trees, and they were all more or less 1,500 years old. But because they were under the conception that big trees are old trees, because that's what was known at the time, they were always looking at the big and healthy trees and ignoring the ones that look half dead. And then at some point, Schulman decided, before they went back, he said, okay, let's give it a shot. And he started to drill into a tree that looked half dead. And the first tree that he tried was over 4,000 years old. And then he understood that he has in his hand like the oldest known living organism. And then they sampled more trees, and they found that there are a number of trees that are older than 4,000 years. And this was published in his paper, Longevity Under Adversity, which basically says that if you grow slowly and you have a hard life, you live long. Okay, that's like, and he proposed a mechanism for this, a physiological mechanism for this. And the sad story is that the person that discovered the oldest living organism on Earth, he died when he was 50. And the oldest known tree on today is Metushelach, and he is 4,854 years old. And this is not him. Okay, they don't tell you who he is. They just, like the people we work with there, they said it's the least impressive tree in the forest, but they were not allowed to tell you which one it is, because Metushelach is the oldest tree today, but Prometheus was like 100 years older, and he was cut down by mistake by a master student, which was drilling and trying to core a few trees, and the drill got stuck in the tree. And this was in the 60s, and back then it was hard to get a new one. So he just went down to the forest offices and asked, "Can I cut down that tree?" And they said, "Yeah, if it's one tree, you can cut it down." And then he took it to the lab and discovered that this was the oldest living thing on Earth, and he just killed it. So this is the stump of Prometheus, and that's why you're not allowed to do. You're not allowed. It's very hard to do research over there because they're very protective of the trees. And I was there a few years ago, and we wanted to look at how seedling establishment of bristlecone pines is affected by ectomycorrhizal inoculum, and how this might be important under global climate change conditions. So a few things you need to know about these trees. So they can live up to 5,000 years, but then they die and they remain standing. The tree that you see here probably died about 2,500 years ago. They can remain standing for 5,000 years after death. I know it sounds very weird to almost everybody, but this is how it works. So, and then they fall over and they can be on the ground for 1,000 or 2,000 more years. So you can actually come across a log that's almost 12,000 years old. And the seedlings grow very slowly. Okay, so this seedling right here is probably 50 years old. Okay, they grow very, very slowly. And because they grow so slowly and because they last in the environment for so long, you can actually see climate change when you take a picture of the habitat. So this is a picture I took. I have to be honest and see that I saw a picture by a famous photographer and it looked amazing, but it was too expensive for me to buy. So I just worked on Google Earth for like an hour trying to understand where he took it from. And then I went and I took a picture from his picture is way better. But what you can see here in the back, these are the Sierra Nevadas, the really big mountain range of California. And this is the White Mountains. And all these things you can see here on the screen, these are dead trees. And all these trees died more or less 2,500 years ago. We dated those trees and that's more or less when they died. And when you look down here, you can see that the tree line, the present tree line is a little bit lower, meaning that these trees established probably when it was a bit warmer. But what you can also see is that young trees are starting to establish above the tree line. So it looks as if the tree line receded and now it's climbing back up. And you can see that those young trees are establishing within these ghost forests, okay, where the dead trees are still there and they're coming back to take their land. So this is a process that's been taking place around more or less 50 in the last 50 years, okay, this climbing of the tree line, okay. So these climbing trees are still relatively young. But when people started to take a closer look, they found something very disturbing and weird. They found that a lot of these establishing trees, and in many places, most of these establishing trees are not bristlecone pines. These are not the very long-lived, famous trees. This is a different species of pine. Okay, this is Pinus flexilis, limber pine, which is also a very nice tree, and it could live for up to like 1,700 years. So it's an impressive tree, but it's not the bristlecone. And so over here we can see a bristlecone seedling, and over here we can see a limber pine seedling, and you can see that they have quite different morphology. And the nice, I don't know if nice is the word for this picture, is interesting because in this habitat, it's very hard to establish in open areas. So they kind of need facilitation. And in many cases, this facilitation is done by abiotic objects like rocks. You see those two rocks here, or just dead trees. So this dead tree is a dead limber, sorry, it's a dead bristlecone. and it's facilitating the establishment of the limber pine. So the question that we were interested is, why are limber pines establishing above the bristle cones, and how is this related to ectomycorrhizal fungi, or whether it is interrelated? And I did this study with Tom, and because both species are obligatory ectomycorrhizal, we wanted to see whether they have differences in the community they interact with. And one of the hypotheses that we had is that these ancient ghost forests that died 2,500 years ago still have a dormant seed bank from 2,500 years ago and are still able to facilitate the establishment of the young trees. So this is very hard to study experimentally. About 20 years ago, Tom started a 100-year experiment. Okay, I just tagged along. Tom started the whole thing. And it's been going for 20 years, and we recently published a 15-year result. And I won't go into detail because I see that I'm short on time. But basically, what we found is that after 15 years, they're still in mint condition, the spores, and they germinate and establish on roots just the same as they did 15 years ago when they were buried in the soil. So we wanted to look at this in the White Mountains. So we collected soil in open habitats next to young sapling, only 50 years old, and next to dead trees. Okay, and we thought maybe next to dead trees will have increased inoculum because of the history of 2,500 years ago. So this was very presumptuous. And then we took these soils and we grew limber pine on these soils, and we grew bristlecone pine on these soils. And what we found is that limber is just better. Limber pine is just better at finding mycorrhiza. Over here, we can see the percent of bristlecone colonized and the percent of limber pine colonized. And you can see that every point is above the diagonal, meaning that for every soil sample that we looked, the limber pine was better at finding mutualists than the bristlecone pine. And you can also look at the species. Once again, Geopera was a big player here. Geopera and Rhizopogon, which is a swilloid. And when we were looking at the different habitats, we found that live trees are the best. When you take soil under a live young 50-year-old tree, everybody gets inoculated, all the seedlings. When you go to a dead tree or to a treeless control, then the inoculation drops, but it drops much stronger for the bristlecones. So the bristlecones are not that good at finding mutualists. And when you look only at Geopera, which was the dominant species, you can see that next to the dead trees, the Geopera is actually better at finding, the bristlecone is better at finding Geopera next to dead trees compared to the treeless control. And this could implement that there is a 2,500 years old spore bank, and that would be very cool, but I don't believe that. And I think that there is an alternative explanation. I think that these old trees, they provide habitat for rodents. And then I think the rodents might be bringing in those fungi, these mushrooms, and eating them next to the dead trees just because it provides some shelter. And then you get a spore bank, but I'm not sure that this is the ancient spore bank. So just to sum up this part, fungi might have a role in the establishment of these trees. And it's not clear if the spore bank can last for decades, but young limber pines seem to be creating hotspots. So we also sampled under young limber pines, and they are creating EMF hotspots. Once they reach the age of 50, there's a lot of mycorrhiza underneath them. And maybe that would facilitate the establishment of the bristlecones. And maybe the limber pines are just early succession trees, and then they die back in the first extreme winter, and the bristlecones can continue their way up the mountain. So I know I am over my time here. actually nobody told me what my time was, but I'm assuming I'm over it. So I'll only take like 2 more minutes to just sum it up on a more global scale. So usually when I give seminars and I'm supposed in the end, think about how this is important and why. So I make something up because I usually do things which are not that important. But actually, when working on ectomycorrhizal fungi, I found a really good real explanation for why this is important and why it's related to global changes. So this is one of my favorite figures. I didn't make it. It's the Crowther lab. They made it, and it shows how organic carbon is distributed on Earth. And you can see a few interesting things here. You can see that most carbon is below ground. Okay, this is above ground and this is below ground. And you can see that most carbon is in the northern hemisphere. I'm assuming it's because they have more land space. And it's in those northern forests. Okay, so this is where EMF rules. Okay, this is Russia, Europe, North America. Okay, this is where these are the ectomycorrhizal forests. And this means that ectomycorrhizal forests overlap to a very large extent with the largest carbon reservoir on Earth, the terrestrial reservoir. So I'm assuming you're familiar with the Gatgill effect because it's a mycological thing, but in general, like saprotrophic fungi, they're responsible for decomposing the soil organic matter. But as long as you have ectomycorrhizal fungi growing, they constantly suppress the saprophytic activity and that means that when we have trees that die because of drought or because of bark beetle attacks, we see a surge of saprophytic fungi, and then we have a surge of carbon dioxide that's released back into the atmosphere. So the trees are the ones fixing the carbon, but the ectomycorrhiza keep it safe and keep it from being decomposed by the saprophytes. So if we want to better understand our Earth's carbon cycles, we need to understand EMF ecology, and we need to understand how EMF is involved in the establishment of new seedlings, because old trees die eventually, even very old ones die eventually. And for forests to persist, new seedlings need to establish. And with that, I'd like to thank you. I hope you're still here because I didn't hear you throughout the whole thing. And I'd like to thank Stav and Ofer and Tom, my collaborators. I want to thank Yael, who drew all these really cool pictures. Connie Miller, which is an amazing forester from the White Mountains, and she was running up all the mountains a year before retirement with a broken arm and helping us drill and collect soil, and all of my other collaborators that helped me along the way. Thank you. And I'll be very happy to answer questions. I hope you have some.
Brilliant. Thank you very much. As always, if people want to pop their questions into the Q&A at the bottom of your screen, then Hagai can answer those. While people are thinking of that and entering that in, thank you very much. That was a brilliant presentation. And going along, I was writing down questions and then you'd answered them, so I'd cross that out. So one, though, I wanted to touch on from your very first area that you were talking about in the boar and the poop. I'm always loving poop stories. So you touched on it very briefly in your last summation slide of that section, but do you have any evidence of whether or not boars are actually facilitating the spread of those forests or the expansion of those forest edges, kind of like what Ian Dickey's group is finding in New Zealand?
So I don't have, well, it's very hard, to establish causality between those two things. But I would also add to that list South America, that Martin Nunes's work. I think they've also, shown that wild boars are moving spores out of the forest, and this at least temporarily correlated with the dispersal of pines outside of the forest. And I think at least it looks like that in our system, it's this Willis, which was already outside of the forest, because we collected in an open field. I'm assuming that wild boars went through it many times before we got there. And I'm also assuming there's different dispersal. There's also aerial dispersal. And for Willis, I think Kabir Pay showed that they can move like a few kilometers by air. But I think that when you have aerial dispersal, the density of the spores is very low. And when you have animal dispersal, the density is very, very high. And at least like, well, from my experience and also from other papers, the spore density is very important for establishment of ectomycorrhizal interaction. And I think that especially when you're talking about such extreme specialists, I think this is more true for pines than for oaks, because oaks have relatively big seeds and they are able to, I think, hang in there until they find the fungi. But at least our pines have very small seeds, and if they don't find their mutualist within a few months, they're dead.
Really cool. So there's a question from Tom May, who is being sacrilegious and asking about the plant instead of the fungus. It's fascinating talk. This is not a question about mycorrhizal fungi, but about the long-lived dead trees. Is this because it is very dry and there's low saprotrophic activity in the dead wood?
So that paper by Schulman, so his hypothesis is that because these trees grow very slowly, it creates a situation where the density of not sap. Sorry, I lost the word. I have it in Hebrew. The word that the thing that turns into amber if it sits in the soil for like 1,000,000 years.
Yeah, that would be sap.
Sap?
Yep.
Okay. So the density of sap, because the glands that excrete, like, is that the word? Like take out the sap, are located at the border between those layers. So when the layers are really thin, the density of sap in the wood is very high, and the density of sap in that wood is extremely high. And also it's cold and it's dry. I mean, there are other places in the US where it's cold and dry, and you can see, I remember I've been to Flagstaff, Arizona, and there's a forest that burned in the 80s and it's still standing. But that's 40 years. So here it's 5,000. So, and now they have a lot of problems of bark beetle in North America. And to this point, bark beetle has not been a problem for bristlecone pines, because it's very, I guess it's just very untasty. And Schulman's hypothesis is that the trees that have it the hardest, that are located in the bad places, meaning that there's less water or more exposure, grow slowly, so their rings are thinner, and that increases the density of the sap in the wood and the concentration of all these materials that are making it harder for microbial decomposition.
Cool. Yeah, I was sitting there thinking, if we're trying to capture carbon, too bad they grow so slowly. Because if they stand up for that long after death, that's impressive. We just have a couple more minutes, so if people have questions, if you still want to pop them in the Q&A, Tom corrected me, it is resin, it is not sap.
Resin, that's it. Thank you.
Yes. So question about the experimentation you've done with the homogeneous versus heterogeneous mixtures. So I was curious about the Geopera as well. So you were talking about priority effect, and I see how that would definitely be a possibility. But I mean, the fact that you have such structured, when you have, the heterogeneous mixtures, and you seem to have really, almost equal portions of all of those, could it also be the fact that the Geopera actually is inhibiting other fungi, but it can only do that at the front of what it grows? Does that make sense?
Yeah, I think that what we found is that, like, when you mix it up, the Geopera is very close to all the root tips, and then it just and I think it's better at quickly establishing, and then once it's established, I think the others have a hard time taking that away from it. But I think that when it's structured like it is here, in some of the cases, the roots grow towards a different species. And then if they hit that species first, the Geopera can't take that away from them, or not to the extent of dominance. Did I answer your question?
Yeah, yeah, it
That's,
does. It does.
I
Um,
mean,
yeah, and
if I was,
is there any evidence either that, so you said the plant root might grow towards one specifically or another, but is there any evidence as well that, that the host might be selecting for the more beneficial? Did you do any single
yeah,
spore inoculations to see if Geopera is just a phenomenal symbiont and giving more nutrients and everything?
yeah. So like I return you to my first slide, which I'm not a mycologist, and I've like I've never done like single species experiments. Not be, I think they're very good in the sense that they have very high internal validity, meaning that you can pinpoint an hypothesis. I'm just don't have a lot of experience like working with specific fungi. But I think that working with forest soil has it really increases the ecological validity of your results, because plants in the soil don't interact with the single species. But I agree that if we were to work with single species, we would be able to, maybe test more specific mechanisms. We were also interested whether the host can move towards, like you said, towards a more beneficial, maybe, and like differentiate between them. But when we did a correlation test, we found that the more Geopera you had, the bigger you were.
Right.
And so it seems like Geopera wins, but it's not necessarily a bad thing for the pine.
Cool, great. Well, thank you so much for today. This has been so phenomenal to hear from you and to hear such great work. And we're happy to have you over here doing your sabbatical as well.
Thank you very much for having me.
And thank you very much to those in the audience and those who will be watching this in future. Again, same time, same place next month. We look forward to seeing you. Bye for now, everyone.
Bye. Thank you.